These are clones. A clone is a living thing genetically identical to another living thing. You and I are one of a kind. But what if you were not? What if we could make a duplicate of you, genetically exact? Should we? People fear genetically engineered clones. Would an individual still be individual? See, it may be possible to use the same techniques that we'd use to clone a new person to instead rebuild or repair organs or even cure genetic diseases. The things we're talking about are microscopic, but the possibilities are far-reaching. Should we pursue cloning? Well, we're going to look at the science, and then we can each decide for ourselves.
The Eyes of Nye.
These are clones. Each of these grapevines is just like all the other vines. They were all grown from cuttings of the same plant. In fact, our word clone comes from the Greek word for twig. See, clones are not that unusual. They're offspring produced without sex. These two plants are genetically identical and asexually reproduced. They both came from a single parent, and they both have the same DNA. Now, when these individuals were created, there was no help from flowers or the birds and the bees. No pollen was exchanged. There was no sex. Ah, humans. Now, we can't reproduce asexually, just cloning ourselves like grapevines. Oh, no. We have to have sex. And for many, that's not a problem. That couple has started on the process of sexual reproduction, where half of the genetic information comes from the woman and half comes from the man. But we're talking about cloning, asexual reproduction, where all of the DNA comes from a single parent. Now, all of the cells in your body that are not sperms or eggs are called somatic cells. And almost every somatic cell carries a complete copy of an organism's DNA. So to clone an animal like Dolly the sheep, scientists start with an egg. Then using the tiniest of glass pipettes, they pull the nucleus of that egg out. And then they get the DNA from another somatic cell. Or they take a whole cell, DNA and all, and poke it back into that egg. Now this is called somatic cell nuclear transfer. It's cloning. Then scientists give this egg a tiny jolt of electricity, or just the right chemical mix to get it to start growing, to start dividing. Then they put it back into the womb of a sheep, and if nothing goes wrong in a few months, you get a lamb. Dolly the sheep was born on the 277th try. That's after 276 failures. Cloning is a tricky business. Now, if we have this much trouble with farm animals, are we really ready to try it with humans?
No.
Proteins in sea squirts and human embryos are the same proteins that are causing embryonic development.
So they're the same genes as people.
Right. A human starts as one cell, a sea squirt starts as one cell. But it's the way the genes are turned on and off that decides what it's going to finally look like.
So you use these animals to study cloning?
Right. We use them to study how genes are turned on and off during development. So most of your cells right now, the DNA is off, which means it's not making protein. They might have been used before to make an ear, but now that you have an ear, those cells are only used for specific functions.
But when you're being born, when you're a single cell?
The egg starts as a single cell, so that first single cell has to somehow turn on and off all the genes necessary to make that adult organism. Now, how does that happen? The egg actually puts proteins in the cytoplasm that tells the nucleus where certain things are supposed to be made. So those transcription factors, proteins...
That's what you call those proteins?
Proteins, yes. Transcription factors will go into the nucleus at certain times and then turn certain genes on and off. And each animal has the same set of genes turning things on and off, but it's how and when you turn different genes on and off that makes different adults. What you do in cloning is you take this nucleus out of there and you put in a nucleus from an adult, and that's the difficult part, is getting that nucleus from an adult to go through this whole process of making an embryo. The reason reproductive cloning doesn't work very well is because you have to take the old nucleus and you have to make it think that it's a young nucleus. You have to make it think it's a nucleus that's in an egg. This is a difficult process, and these nuclei are old nuclei, so a nucleus in your, say, skin might have lived so far for a number of years, maybe 40-some years, and so you have to take that old nucleus and try to make it young again, and we're not sure how to do that. That's one of the most difficult things about reproductive cloning. It's a very low success rate, maybe 1% to 2%. If you try to clone yourself.
Is that why Dolly was such a big deal?
Well, Dolly was a big deal because she was cloned. She also died at the age of 7, which is about half as old as a regular sheep would live. And so there's some idea that the nucleus was too old, and that's why she died at a premature age, is because even though the nucleus was tricked into thinking it was young again, it really wasn't that young again, and so she died.
When it comes to the technology of cloning, the train has pretty much left the station. See, once we've performed somatic cell nuclear transfer, we get an embryo, not a fetus, an embryo, cells that are dividing. Now, there's two choices as to how we use that embryo. We can go this way. It's what we call reproductive cloning. Make a new cow or a sheep, or theoretically, even a person. But we don't have to go that way. If we don't implant the embryo into a womb, just let it grow, we can instead go this way. And this is therapeutic cloning. And the cells grow and divide. It's what we call stem cells. Now, stem cells can become any type of cell. And here's the thing. The procedure for reproductive cloning and therapeutic cloning is exactly the same up to this point. Up to this point, this fork in the road. If we go this way, we would get new people. If we go this way, we would get new cells. Which way do you want to go?
We've covered a lot of ground, but it's just the tip of the iceberg. Check out eyesofnye.org for more cool science.